Okay, our next speaker here right after the break is Dr. Mark Thallman, who needs little introduction, I expect, to most of you, but research scientist at US Meat Animal Research Center and former BIF genetic prediction, genetic, we changed the committee name enough times, but genetic and genomic prediction committee chair. Mark's talks a vision for the future of low-pass sequencing. Dr. Thallman, don't forget to turn it on.
Okay, I appreciate the opportunity to speak to you today. I want to try to challenge you to think about low-pass sequencing in a little bit different way, and to really think about what should it look like 10 years from now, instead of, I'm not going to talk to you about how we get from here to there. All right, so kind of an overall outline of what I want to cover is some of the constraints that I've heard in the past to the widespread adoption of low pass, how to represent genomic sequence efficiently, how to impute low pass more efficiently when both of the parents already have imputed phase sequence. Want to propose a completely different way of thinking about the genomic prediction part of the problem that involves a hierarchical statistical model to utilize genomic sequence, and that's motivated by biological processes and molecular biology. It'll look pretty different from what you're used to seeing. I'll discuss some other more ambitious opportunities at the end of the presentation. And I'll just say up front that this is quite a bit of stuff to cover, and I am intentionally glossing over a lot of details that I've already thought about, and I'm sure that there are a whole lot more details that I haven't thought about. In contrast to the other people who spoke in, I haven't done any of this, but I've been thinking about it for longer than most of the ones that have done. So, like decades. I have been thinking about it for a while. So the constraints that I hear are, they basically center around cost and how much is the added value of doing low pass. And the main impediments I see on cost are the cost of collecting a tissue sample to begin with, the cost of DNA extraction, constructing barcode-coded libraries for sequencing, the actual cost of running the sequence data, and the cost of storing the resulting information from it. And in terms of value of information provided, we've got several categories there. How much are we going to improve the accuracy of predicting breeding values for use in selection and mating? Potentially, can we add enough value to make it work for marker-assisted selection so that there's a good value proposition for that? And then a central thing that I hear is concern that adding more variance is not going to translate into greater accuracy of predictions. So I'm going to try to address not every one of those, but most of them. So to start out, I want to challenge you to think about, there's roughly 100 million cattle in the U.S. Let's just suppose that we had genomic sequence on all of them. And we wanted to store the information contained in that sequence in such a way that we could reconstruct that sequence for any one of those 100 million animals. How much storage would we need to do that? And so I'll walk you through how I look at the problem. And so it starts with taking the genome and breaking it into a number of defined segments. And then in each one of those segments, we need to know what's every haplotype for that defined segment that exists in the population. And we're going to enumerate those with an integer. And I'd like for those segments to be however short they have to be, that we can do that with no more than 255 haplotypes, and you can probably target less than that, because that number could grow. 255 is because I want to be able to store those in a byte. And then I don't want those segments any longer than 32,767 because I want to be able to store offsets into those segments in 2-byte integers. Okay, so this is a start of a schematic. So we're going to start with a, I'll call it a reference haplotype. And to the extent that we can do it, it'd be one that's represented in the population, but to the extent we can do it, it'll be the root of the phylogenetic tree. So we have descendants, and for each descendant, we're going to only store the offset into the segment and what the substitution is. And on this last one, you'll see that our phylogeny didn't work anymore because it's actually a chimeric haplotype that starts out as haplotype 8 and finishes up with haplotype 4. So we've got to treat those a little bit differently, and those chimeric haplotypes cause our phylogenetic tree to no longer be a tree but to have loops in it. So the reference sequence, if we go across the whole genome, requires approximately 3 gigabytes of storage across all of the segments. So if we assume that there's 250,000 segments with an average of 200 haplotypes per segment and 2 new variants per segment per haplotype, and it takes 5 bytes then per new variant. And so that implies 500 megabytes to store the variants. So the way we store the sequence of individual animals is to represent these reference haplotypes across the genome segments. And so for each animal, we've got a haplotype for the paternal chromosome and a haplotype for the maternal chromosome. And that's going to be 1 byte per segment times 2. And this is conceptually how we want to represent the data. It's not how we'll actually store it, but it's a precursor to the way that I want to use the information in the genomic prediction model. So the way we're actually going to store it is starting from the bottom. We're going to start out with, at the beginning of a chromosome, do we have the maternal, the grandmaternal, or the grandpaternal haplotype for each of the maternal and paternal haplotypes. And then we have the segment number at which the first recombination occurs, and the offset into that segment, and the length of the region of ambiguity. So how far is it until we know that we're sure we switched to the opposite parental origin. And, we just keep working up through the pedigree that way. And then when we get to founders, we still need to represent the series of actual haplotype identities. So we assume we've got 250,000 haplotype segments. I'm going to assume that that's going to take 500 kilobytes per founder, and that we've got 500,000 founder individuals across the beef industry. That's going to add up to 250 gigabytes of storage. And then for the more compact storage, we've got 60 chromosomes, and we'll use a byte on each of those to determine whether they started with the maternal or paternal origin. And then we expect around 60 crossovers per individual. And each time we have a crossover, that's going to take 10 bytes. So there's 600 bytes per individual. We're going to have mutations and other things, structural variants, copy numbers that we got to account for. So let's round that up to a kilobyte per individual times 100 million animals is 100 gigabytes. So 4 plus 250 plus 100 is 354 gigabytes. We can store that on a micro SD card. It costs $33. And you can stick it in your phone. And if you got the right software, you can pull up the sequence on the animal you want. So next problem, how do we save on the cost of doing low pass. And for me, the key to that is that we need to implement an algorithm to impute low pass from parents that already have phase sequence. Now, I've heard that that's too big a headache to find out who the parents of the animal is when it's submitted. Doesn't work for animals with unknown parentage, but the advantages of doing it are very substantial. So I'm going to go through it with you. So our current low pass algorithms impute each animal individually without considering pedigree. But it's much more efficient if both parents are known and have imputed phased genomic sequence. We'll be able to lower the depth of sequence coverage substantially, and we'll also have less computational expense. So how do we do that? We've got here on the slide, we've got the phase sequence of the sire, the phase sequence of the dam, and then reads on the progeny. And here I'm indicating on these reads with the color whether each of those reads came from the dam or from the sire, and also which of the haplotypes, because there's no crossover within this region. So all of the ones that came from the sire came from the maternal or the paternal grand dam. So this blue haplotype across here, they shouldn't have come from this green haplotype, but I've got one read here that is assigned to the green haplotype because that's what its sequence matches more closely. And that's due to a sequencing error. And those are going to happen. And they're going to happen enough that it's not trivial. And then the rest of them are red, and that indicates the grand paternal haplotype here in red. And so other than that, other than sequencing errors, we shouldn't have any that are green or purple here. So we got one green due to the sequencing error. Unfortunately, reads don't come off the sequencer painted by grandparental origin. So we have to look at them as unknown, and we have to use the sequence of the reads and how that sequence matches the 4 grandpaternal haplotypes in order to try to infer where they came from. And there are 6 of them here that are bolded and colored for which that's possible. And what that means is that the sequence of that read matches only one of the 4 grandparental haplotypes. If it matches more than one, we consider it uninformative. There is a little bit of information there if it matches one of them, but not, yeah, sorry, I'm not thinking of how to say that right. There's a little bit of additional information there, but for the purpose of the presentation, I'm going to assume that what I described is enough to get us where we want to go, and I think it is actually. So we're going to transform that information into these rows of 1s and 0s. I've got question marks there for the reads that weren't informative, but the important thing there is the 1s and 0s. They look pretty sparse at this scale, but we're looking at a really tiny piece of the genome. And so what happens next? We look at a bigger piece of the genome, and the grandparental origins down here look a lot more dense. We still have some here that are highlighted that are out of phase with the ones surrounding them, but we know we don't have that many crossovers in a little tiny piece of chromosome. So it's pretty easy to figure out that those are due to sequencing errors that just cause them to match better to the wrong haplotype. We do have a crossover here in the area that's green, and so there's a little region of ambiguity. So we know up to this point that we're maternal. We get over to here and we know we're paternal, but we don't have any reeds that are informative within this region. But that's generally going to be a fairly small region, but it's worth keeping track of. Okay, so we can push this up to a larger scale look at the chromosome. And so what I'm representing here is that this region down here corresponds to a small area of this second segment, 902. And that allows us to reconstruct the haplotypes that I showed you for this animal on the previous figure. And then if we move on up to a large scale, you can see that most of it's going to be imputed with near certainty, and we just have these regions of ambiguity, and these sections here in the third section are locations of crossover events. So another concern is, what are we going to do with all the reads that we generate? And so the first thing is, we're going to be comparing those reads. So we're going to map the reads to our reference haplotypes. And I think I forgot to add to this, if we have any reads that don't map to our reference haplotypes, we want to put them in a bin and store them. And eventually that's going to allow us to find variation that's outside of the core genome and other problems like that in our reference that we use to map the reads in the past. So, so we'll store the unmapped reads. For the reads that are mapped, if they match an enumerated haplotype within a reasonable amount of sequencing errors, then there's no need to store those. We just know that that animal at the version of the reference haplotypes that we're dealing with, that animal and a sequence that is sorry about that. That animal is assumed to have the sequence that was represented in that reference haplotype. If we improve that reference haplotype in the future, that animal sequence will be automatically improved as well. So the reads that we really want to store are the ones that are most likely to lead us to new reference haplotypes. And so we need a way to accumulate counts of how many discrepancies have occurred at each position of each haplotype of each segment. And that'll take a bit of storage, but it's not unreasonable. And then we rank positions, and those that have the greatest numbers of discrepancies are the ones that we save reads for, so that we can go back and use those reads in a process of continuous improvement of our reference haplotypes. And in the long run, the improvement of reference haplotypes is going to come from sequence reads that are generated from low pass. We're never going to deep sequence enough highly influential bulls to span the whole population. And so there's probably a need to do a bit more deep sequencing, but in my opinion, it would be fairly limited. And in breeds that haven't been well enough represented so far. A lot of the ones that aren't discovered are going to be at low frequency in the population. So not having them discovered is, in the short term, not going to be a huge hit to accuracy of prediction, but in the long term, we want to find them. And in the long run, we'll find rare variants that will have important effects. And as I said earlier, when we improve a reference haplotype, that's automatically going to improve the sequence of the individuals in the population that have been assigned that reference haplotype. And over time, this sequence generated from low pass that's going to start in one animal, move up the pedigree, and come back down the pedigree to a lot of other animals is going to reduce the size of these regions of ambiguity. So those regions can be big when they first occur, but as more and more animals that are descended from that crossover event are sequenced, those regions of ambiguity will get smaller. Okay, so I'm going to propose that we really need to have two different low pass products. So the first of those is what we have right now. And it works on any animal, pedigree known, unknown. Don't have to worry about sequence on the parents. It'd be what I consider founders. I'm sure there's room to improve the current product, but those would be incremental changes. But the second product would be specifically designed for progeny of genome imputed parents. And it's going to be a pedigree-based imputation, as I just described. That's going to require much lower sequencing coverage depth. Probably that can go about as low as you want to take it. You know, if it's a choice of spending 5 cents an animal on sequence or 25 cents or 50 cents, you know, you can make that decision. But that's the amount of sequence that we ought to be looking at applying to this problem, because all we have to do is determine did the chromosome start out maternal or paternal and where are the crossover events. And it just doesn't take that much sequence data to do that. And, you know, the only real benefit of more, well, there's two, I guess. One benefit of more sequence coverage is smaller regions of ambiguity. The other one is more sequence data to go back and improve the reference haplotypes. And that's all I see as advantages from more coverage. I will point out the computations are going to cost less if you have less coverage. And then we need a different protocol for barcoded library construction that's optimized for this purpose. And a real limit right now on the cost of low pass is what it costs to build that library. Am I right, Stuart?
It's a piece of it. It's a piece of it. There are ways to minimize that cost, but it comes at a cost.
Yeah.
But yeah, it's a consideration for sure, Mark.
Well, we need to drive that cost down for this purpose. And really it should minimally cost just a bit more than a PCR reaction. And I think there's a lot of advantages to doing it in such a way that it gives us a reduced representation of the genome, and that is emphasizing the regions that flank genes. So we do a really good job of imputing the origin of genes and not so good in genes. The advantages of all this are a lot greater if an entire herd or population is sequenced than just a select few, so that at each generation, once we've got the founders done with the more expensive product, we can use this less expensive product at scale. And it's going to be important that it's able to run at scale, because we might be looking at 10 to 50,000 animals per flow cell on your high capacity sequencer for this. And so we need to have a lot of scale to really drive the cost down. So we need to drive the cost down to get to that scale. I think it's achievable. Um, that's it. Okay, so that's kind of it on how we do it. And I want to spend the rest of the presentation on how could we achieve greater benefit from low-pass sequence than from chip genotypes. And a big part of that is going to be a hierarchical statistical model to utilize genomic sequence. It's motivated by biological processes in molecular biology. It is, you'll see, very different from a linear additive model. It's multiplicative instead of additive at several levels of hierarchy, and that's motivated by the biology. And it's gene-based instead of SNP-based, and we have been ignoring an enormous amount of information that's readily available to us in our current genetic models when we treat every SNP, regardless of where it is in the genome, as being equivalent to any other SNP. And I'm going to suggest that that needs to stop. This approach is designed to severely reduce the overparameterization problem and basically to reduce a lot of places in the model where we have numerous individual SNP effects with one effect per gene, and that occurs at a couple of levels of hierarchy. And then it's going to lean very heavily on information that's external to the genomic evaluation to estimate some of the parameters in the model. And also use external information for feature selection, driving how we improve the model, and a whole lot of other things. So the starting point is a gene, and what I'm trying to represent in this diagram is that right through the middle here in this gene model, we have vertical bars, and those represent variants. They could be SNPs or other kinds of variants. And these variants come in three different kinds. The ones in purple are variants that influence the expression of the gene. And they are also represented on this level up above. The ones in green affect gene function. And I'm going to call it functionality because I mean a number, not does this gene work or does it not work. I mean a number. So I'm just going to use a slightly different term for that, functionality. And those variants are represented below. And then the ones that are black up here are variants that have no effect on either the expression of the gene or how functional its product is. So we have three classes of SNPs. And in the biological model, we know what those are. You notice I have a few of the functionality ones up above because we don't know that there aren't some variants that affect both gene expression and function. And so our model is going to be based on a haplotype of the expression variants and a haplotype of the functionality variants. It's going to be derived from the haplotypes of the genomic variants, but what we're going to use in the model is only, to the extent we can identify them, only those variants that affect functionality of the gene for one haplotype, and only those variants that affect expression for the other haplotype. And so we get a set of expression haplotypes here, functionality haplotypes here, and then for each of those, we want a set of estimates of what the what the expression is for each of these expression haplotypes, and what the functionality is for each of these functionality haplotypes. And for functionality, that's going to be estimated from an information source that's the trait phenotypes. But for expression, I'm proposing that to the extent that we can get it done, and we don't have the data today, I think, to do it, but we're going to use RNA-seq. And, sorry, I'm not there yet. I'm going to explain later how we'll use RNA-seq to estimate the expression associated with each of those expression haplotypes. Okay, so the biological model for how genes affect phenotypes starts here. And what I'm representing is that over here, we're looking at gene expression from the maternal chromosome, and then we have gene functionality from the maternal chromosome. Those two don't get added together, they're multiplied by one another. If either one of those is zero, the result is zero. Okay? And that happens, we either get functional gene product from the maternal chromosome, and it can vary in amount, or we don't. And then we have a parallel process happening from the paternal chromosome. And so we have the paternal gene expression, the paternal gene functionality, we multiply those together, and we add that to the maternal gene activity, and I'm going to call that individual gene activity, represented as AJ, where J is a subscript indicating what gene we're looking at. And I want to point out, you could have a functional maternal, you could have expression of a maternal gene that's not functional, as a knockout. You could have a complete lack of expression of a paternal gene that's perfectly functional, and you still have zero gene activity. And in that case, for the things that are non-zero, it doesn't matter how non-zero they are, whether they're a little bit non-zero or a whole lot non-zero. It's just irrelevant. We do have parental imprinting at various locations in the genome and various tissues in the genome. And this is what would happen if we had parental imprinting, is we would just have the right-hand side of that graph. We could equally well have maternal gene, or shouldn't have said paternal imprinting, paternal expression, or we could have maternal expression, and that would be the other side. So how does dominance occur? Well, there's several different models of dominance, but the one that I first learned, and I think is still probably the one that operates most commonly, is that the relationship between gene activity and gene effect is nonlinear. And I'm representing it with this curve. And so for this simple two allele gene, it's really easy to figure out that the difference between the curve and this line segment between the two homozygotes is dominance. And the part from here to here is additive. Kind of makes sense, but I need to define a little bit better for you what I mean by gene effect. And so imagine for any gene that we had some quantifiable measure like a lab assay that said, what is the effect of this gene on phenotypes? It's not necessarily going to be the same for every trait. I meant by trait phenotypes. Not necessarily going to be the same for every trait, but let's assume that there's a common component of that. And that's what I want this to be. So for example, for a gene that produces an enzyme, the measure would be enzymatic activity. For a structural gene, it might be just the amount of protein that's produced. And this gene effect measure takes into account the negative feedback that's common in molecular biology, so that the more gene effect that is produced from a gene, you have downregulation on the expression of that gene, and that is one of the ways in which this curve can occur. One of several ways in which this kind of a curve can occur. And so ideally, phenotypic variation in any trait influenced by a gene would be proportional to this gene effect. Not always going to happen, but it's one of the simplifications of this model that I mentioned earlier. Okay, if we have multiple haplotypes, then we can see some interesting things here. So haplotype A obviously had the smallest individual gene activity. It actually had 0 for gene activity. And B then was a bit higher. So if we're at the low end of the scale for gene activity, we see a lot of dominance. When we get to the high end of the scale for gene activity, there's no dominance, but there's also no real change in what the gene effect is. So we're reaching the asymptote of the curve. But for the model, we can't look at those haplotypes like that. We need to look at what is the gene activity of an individual at a gene, and we just have the curve. And the curve that I chose here is what's essentially a Brody curve with a birth weight of 0. So I mentioned that the dominance model that I described there with the curve is not the only one. So we could have an additive relationship between gene activity and gene effect. And that is represented by this term. And then this is the term for the curve that I just showed you. So we're going to estimate how much of, where does it lie on the scale between additive and that curve. And then there's another model for dominance that says that if you had a dimer protein where having dissimilar components of the dimer is advantageous, like could happen in some immunological genes, then we can fit this model over here. And it basically just imposes a penalty on haplotypes that are homozygous. Okay, and then from the gene effects, we're going to translate that into a vector of total genetic merit for each individual. So that's going to be the sum of the gene effects for each gene. And then we have a term over here that is 2A epistasis. And we'll estimate the effect of that. It's a multiplicative model. And what I want to emphasize here is this is a scalar parameter, which is what we want to estimate. So we're using the product of gene activities, gene effects, and we're estimating the scalar product, the scalar, so a scalar per interaction between genes. And that's two-way interactions. And it's not as explicitly indicated here as my previous version of the slide, but basically we're not going to look at all pairs of genes as we're doing this. We're going to look at genes that are in the same biochemical pathway, genes whose products bind to one another, or transcription factors and genes that they bind to, places where we have a reason to believe that an interaction might occur. And then where we find pairs of these that have a gene in common, will fit a three-way interaction. And we could go further with that. So this is what we've discussed. This was one slide, this is another slide, this is the next slide. And then in the end, we're going to take the UT from the last slide, and we're going to fit it in the standard genetic evaluation model, standard mixed linear models. I'm not going to go through all of the numbers of how many parameters we have to estimate to do this, but it is a lot less than fitting an effect for every SNP that we find in the genome. So I mentioned earlier that we want to estimate the effects of our expression haplotypes from RNA-seq data. And this slide goes through how to do that. it basically starts with a GWAS that considers only SNPs that are in and around the gene that we're looking at expression of. So we're doing an independent GWAS for every gene, and that greatly reduces the overparameterization problem that we typically have in GWAS. That's going to lead us to a reduced set of variants that we're going to consider to construct our expression haplotypes from. We're going to compute the phylogeny of those and use that to construct a covariance, the structure of a covariance matrix for these haplotype effects, and then that will give us the estimates of expression as a function of expression haplotype. A lot less parameterization than what we typically do. And it has an additional advantage. So first of all, those expression haplotypes might account for roughly half of the parameters that need to be estimated in this model. And so using an independent source of information is useful for that. Also, if we're trying to estimate gene expression of haplotypes and gene functionality of haplotypes by multiplying those results together and then feeding that into the rest of the hierarchical model, those effects are going to tend to be fairly highly confounded with one another. And so having an external estimate of one of those makes it a lot easier to estimate the other one. And so, in fact, that first equation in the hierarchical model that looked pretty nasty becomes just a simple linear mixed model when we take the gene expression effects and use those to calculate the design matrix for the gene functionality effects. And I'm not going to show you the math for that, but it works out. So this hierarchical model is probably easiest to fit in the short run with MC-MC because of all the nonlinearities and kind of complicated terms. I think the conditional sampling distributions for some of those terms may be more complicated than for a strictly linear model. And we might have to lean on Tropos-Hastings quite a bit to do that. In the longer term, I want to explore using non-stochastic conditionally linear mixed model computations to run these models. And I think there's some opportunity there. I'm not sure that it'll work, but I'd like to give it a try. So there are a lot of assumptions in what I've described to you. And so one of the default assumptions is that the effects of haplotypes on gene expression and functionality are proportionally similar across traits for the same gene, and that the same is true for the dominance net stasis effects. I think we know that's not true. It's a simplifying assumption. And I'll describe a couple different ways that we could deal with that. The default assumption is that haplotype effects on gene expression are proportionally similar across tissues, physiological states, treatments, et cetera, environments. And so the long-term approach to that is fit random effects that account for the departures. So use the common component as kind of a starting value, a prior, a mean in the other model. And then where we have evidence of departures from that, we use the random terms to estimate the departures and account for them. In the shorter term, the first thing we could do is we could just fit the model that I proposed and then add a G-BLUP term to it and let that account for things that the other model didn't account for. And then there's a whole lot of things that we could do that are intermediate to expand on that as well. Okay, so that kind of wraps up what the statistical model is. I want to talk about, if we get that to run, what are things that we could do with that in addition to what we currently do with genomic prediction? And so I've kind of proposed over the last year a couple of different ways in which I thought that we could impute RNA-seq measured intensively on small reference populations that have, from which we can collect a lot of different tissues into larger populations. Also, RNA-seq on large populations, not really large, but larger than what we typically do RNA-seq on, and impute that to much larger populations of animals that have genomic sequence. And as I've been proposing these things, my colleagues have been sending me papers. And I haven't had time to read the entire papers, but I've looked through the abstracts and introductions of them. And they basically describe doing in humans what I've been proposing to do. And so this is ambitious, but I want you to think about what the possibilities are if we move to low-pass sequencing. So I'm going to use the germplasm evaluation population at US-MARC as an example of a reference population. I personally have the highly biased opinion that it is the best reference population to do this with that exists, but other populations could be used as well. So we're going to start with a subset of the GPE population that we can probably harvest in our meat lab and collect a very large number of tissues from on each animal. We're going to start with those animals are already going to have routine phenotypes that are included basically in national cattle evaluation. They're going to have whole genome sequence. They're going to have intensive phenotypes that we collect at US-MARC on GPE that are not routinely collected in national cattle evaluation. They're going to have RNA-seq from blood. They're going to have RNA-seq from a lot of other tissues as well. And then they're going to have other omics, and that could be proteomics, metabolomics, metagenomics, epigenomics, and I just heard of spliceomics or something like that. It gives an RNA splice. That was a new one to me, but we should do that one as well because I think it's really important. All right, and so we should be able to impute that to the rest of GPE. And there are papers saying essentially that in humans they're doing this. I can't understand when they talk about what percentage of genes work. I can't interpret that. I need a correlation. But anyway, I'm pretty confident that this will work. All right, we want to expand that to a bigger population. And from my perspective, that bigger population is our national cattle evaluation populations that are genotyped. Okay? And these papers say that whatever we did that came from up here could come to here. But I'm pretty confident that if we can impute the ones from here into here, and then impute that to here, we can do better. Okay? And I know a lot of you have heard our GPE presentations ad nauseum, some of you haven't. I just will emphasize that we go to great lengths to sample sires that are as highly representative of their breeds and that represent their breeds as fairly as possible is what we can do. So I think that makes this a pretty good population to draw this connection here. And I think that in the end, this information feeds back both ways. And one of the real opportunities I see from doing this, well, let me just be more explicit about what, I'm going to go one more step. The other population that we have, so let's say this is millions of animals or 1 million animals. And then we got tens of millions of relatives of those animals that have the, that have the NCE traits on them. Well, that gives us a lot better phenotype to work with on these animals that have genotypes. Essentially a de-regressed EBV on them that takes into account the phenotypes of tens of millions of animals. Okay? So, how does the information flow from GPE to here? Well, it means that we could be looking at imputed gene expression across tens of thousands of genes times however many tissues we have over here that we're able to do that with. And I would argue that having a big part of the population where we have RNA-seq on blood together with a small part that has RNA-seq on lots of tissues is going to get us a long way down the road to doing that. And you might think, well, that's a daunting problem. How are we going to analyze that? Well, that's a problem I'm interested in. But what's the good of it? Well, we're going to have genetic correlations between our economically important traits and gene expression for some genes. It's just got to happen. And when we fit a prediction of the economically important trait that's conditional on a large number of correlated traits, even if individually their correlations are low, that is going to substantially improve the accuracy of genomic prediction. So I think that's a really huge opportunity. The other thing that happens is we've got this arrow that goes from here to here. And think about what you could do if you had those genetic correlations based on millions of animals of traits with gene expression in a variety of tissues. That generates an enormous amount of information to go into what's called system biology. Yeah. And I know that the human and mouse communities have way more resources and way more data than we do. I don't think they have anything that would parallel that. I really don't. And so I think that is an enormous contribution to science, but it also feeds back, it allows us to do work at US-MARC. It's more targeted. Where are the gaps in the information? What are things, how do those relationships work in the traits that are not included in natural cal evaluation? And then that information feeds back here. So I just think that's an enormous opportunity. And we're not going to get there without doing low-pass and replacing our SNP genotyping with that. Yeah, and where I think there actually is an opportunity. Well, I'm going to pass on that one and move on. Okay, so the other one is opportunities for genotyping large numbers of commercial cattle for marker-assisted selection. Now, marker-assisted selection is what we used to call it. It's part of what's now precision livestock management. I'm pretty sure we'll get more money for it if we call it precision livestock management. But this is not something that hasn't been tried, and Donna's going to talk to you about it tomorrow, I think, on a really large scale. But, you know, Cargill actually tried this 10 or 15 years ago and genotyped 600,000 cattle into feedlots at considerable expense per animal. And the problem was that the benefit that they could get from it wasn't enough to justify the cost, or they'd still be doing it, I think. But we're a long way down the road to being able to provide more value. And the potential exists to get a lot closer to making the cost manageable. And so at some point, those two lines are going to intersect. You know, we probably have to go past the point where they intersect, people to adopt it. But I think it's going to happen. And there's really important feedback loops involved in all of that. And one of those is on the cost side. If we can ever get to the point that this is done at large scale in feedlots, then we're not going to have to worry about how long does it take us to get enough samples to justify doing the sequencing. And that'll be a big help. And as that happens, that will drive the cost per animal of doing this down. You know, another thing I forgot to mention earlier on the less expensive product, that has got to be a product where if you send one sample in, you're going to pay through the nose for it. You know, those samples need to be arriving in large batches so that they can be handled efficiently. And that has to drive the cost. But those things reinforce one another. And the other feedback loop is the value of the information provided. So we're going to have to initially do marker assisted selection with the information that we have when it starts. But that can very rapidly overwhelm the information that we have now, the phenotypic information that we have now, for the traits that are relevant in that production system. And assuming that that data is not lost, and that's not trivial, that can greatly improve the predictions that we have for using seed stock. And having information that's produced in a commercial setting actually doesn't just improve the accuracy of the predictions that we have for seed stock, it improves the relevance of the predictions that we have for seed stock. Because if we're really breeding to serve the commercial industry, traits as they're measured in commercial production are much more valuable than traits measured on seed stock. So I think that this opportunity feeds in really nicely with the imputation that I described and moving a lot more information out to national cattle evaluation populations. And so I think there's really beneficial feedback loops between those two opportunities as well. Final opportunity I'm going to describe is that I've got a postdoc position open to work in variance component estimation. And if you would be interested in that or know somebody who would be interested in that, please let me know. This is going to be focused especially on improving the computation of variance component estimation, but there also are things I want to do in terms of getting more out of the analysis. And with that, I'd like to thank my collaborators. Bailey Engel helped me tremendously with getting this presentation together. And I promise you, it looks much nicer than if I had done it by myself. And Bailey is the newest scientist to join our genetics group at US-MARC. And she's back here in the back. And if you don't know Bailey, introduce yourself to her and get to know her. Bailey, Larry, King, Warren Snelling, and Brittany Keel all went through a very preliminary draft of this with me and all gave really great suggestions to make it better. And I want to thank them as well. And with that, I'll wrap up. And are we taking questions now or going straight to the panel?
Let's just go straight to the panel.
Okay. I don't think.
I thought so.